Welcome back, dear students. Today our topic is genetically modified organisms or transgenic organisms. First of all, how you can define a genetically modified organism? Any organism whose genetic material has been altered by the introduction of a foreign gene or a number of foreign genes from a desirable organism constitutes a genetically modified organism or transgenic organism. Say, for example, we have two plants, plant A and plant B. Plant A is high yielding, but at the same time it is susceptible to a disease or an insect, and plant B is low yielding, but at the same time it is resistant to a disease or an insect. The resistance against disease or insect is because of the presence of a particular gene in species B. Okay, and now you wish to insert this gene which gives resistance to species B into the genome of species A. The procedure involved, the procedure that helps you to take this gene from species B and insert it into the genome of species A, this whole procedure is known as recombinant DNA technology. Okay, so you can say recombinant DNA technology is the procedure by which a gene or gene of interest or a desirable gene is removed from one organism and introduced and inserted into the genome of other organism, so that the later organism also gets that character which was present in organism B. Okay, at this point of time I do not feel that you are required to go into the details of recombinant DNA technology because it is not in your syllabus. Apparently, it seems quite simple to transfer gene from one organism to another organism, but in reality, it is quite cumbersome and tedious to do all this procedure. Okay. Still, we will have a look on how this is done in the laboratory. Say, for example, you want to transfer a gene into your crop. Okay, and here in this example, the gene is known as BTG. और ये जीन जो है it is present in a bacterium known as Bacillus thuringiensis which gives resistance against insects. The first thing that you will do is you will remove this gene from the genome of bacterium. Okay, you have got the gene and place this gene between the promoter and terminator which are specific for maize. Okay, आपके पास एक crop है maize. Okay, you want to have that maize which is resistant to insects. Okay, you will get this gene from the bacterium and insert this gene between the promoter and terminator which are specific for maize, and finally the whole fragment of DNA, it will be introduced into a plasmid. What is a plasmid? It is an extra chromosomal heredity determinant which are present in bacteria. Okay, now this plasmid will be introduced into the bacterial cells, and you have got a bacterial cell with a recombinant plasmid, and this bacterial cell will be placed in the culture medium and it will divide and produce a large number of bacterial cells, and all those bacterial cells will have this recombinant plasmid inside their bodies. Okay, so you are multiplying the gene of interest. Okay, and finally when you have got enough number of these recombinant plasmids, the final step is how you can introduce this DNA into the genome of target cells. Say for example, यहाँ पे आपने एक काम किया, आपने एक प्रोटोप्लास्ट लिया मेज का. You have taken a cell or a protoplast from maize plant and allowed this protoplast to grow here in culture medium. Okay, then what you will do is you will try to insert the gene of interest into the genome of this protoplast. There are a number of methods that are used to insert the gene of interest into the genome, but at this point of time you are not required to know about all those methods. आपको मैं एक example बताऊंगा. One method of introducing the gene of interest into the genome of target cells is known as particle gun method. Particle gun method में हम क्या करते हैं? The gene of interest is coated on small particles of tungsten and gold, and these particles, they are accelerated to move into the target cells, and once they get into the target cells, they enter the target cells. By chance, a few of these coated particles, they reach the nucleus where they exchange the gene of interest with the genome. Okay, and by this, the gene of interest gets introduced into the target cell, and you have now a recombinant cell. And then this recombinant cell, it is allowed to grow to form the callus, and this callus is finally subcultured to get a lot of callus, and finally the callus may be allowed to form the roots and shoots, and you will get the plantlets. Okay, and all the cells of this plantlet, they will be having the gene of interest inside their genetic material. And this is the way how you can create transgenic crops. Okay, this is all about the procedure of recombinant DNA technology, and in the next part we are inshallah going to discuss the BT crops.